This is the lecture about immune system, chapter 21. So over here, we're going to describe all the different limbs of our immune system that we have and different kind of protections that they give. We're going to start off with the classification of the immune system. There are two distinct groups. One is called as innate or non-specific immune defense system, and the other one is adaptive. Now, the difference between these two systems is how specific is the attack towards the antigens. How specific should the antigen be in order for our immune system to protect us? The first one, obviously, the innate defense system has two branches. One is the first line defense, where you have your surface barriers, and the other line is your second line defense, which is the internal defenses that we have. Now, in general, the innate defense, both of them, they do not distinguish which antigen they're going to fight against. Anything that does not belong to our body, this innate defense will attack it. So that is your innate defense. The third line or the last defense mechanism that we have is adaptive defenses, which is more specific. It has memory, and it's basically system-wide immune system that we have. And there are two categories of it. It's your B cell and T cell, or humoral immunity, and your cellular immunity that we have. Now, this adaptive defense, again, the foreign antigen has to be in the blood or in the tissue in order for these two to attack. So these are the two broad classification of our immune system that we have. We're going to look into each and individual defense mechanism. Starting from the first line defense mechanism, we have the surface barrier. The surface barrier includes the skin and mucous membrane and all of the secretions along with it. So it's a physical barrier, and it repels pretty much everything that does not belong to you. It also helps in chemical resistance, and it binds to certain things and makes it inactive. So let's look at all the chemicals that these guys produce. We know that skin in general itself is made of multiple layers of stratified squamous epithelium. So you have that. The topmost skin layer peels off because they're dead tissue. It's constantly slough them off of our body. So that's one of the things. But you also know that the skin has multiple glands. So it has sweat glands, it has acid glands, those kind of things as well. So, not acid, sorry, oil glands, and these secretion actually helps to produce an acidity on our skin. And this acidity keeps the count of our microorganisms on our skin to its minimum level, that it doesn't cause us any disease. In mucous membranes, you have enzymes that are released, like in salivary gland that releases in the oral mucosa, the respiratory mucus, lacrimal fluid, the acids in the stomach. Those are all helps to kill the enzymes. Like in saliva, you have lysozyme, and also in your tears. Mucin in the mucus helps to become sticky, and it traps the microorganism, and then you can kind of expel it out. You also have other things like defensin, which is an antimicrobial protein, and dermacidin, which is also another chemical that can kill the bacteria. The respiratory system, the mucus lining in that system, the lining itself has cilia, where it produces mucus, and then it traps the mucus, and the cilia actually traps the microorganism in the mucus, and the cilia pushes it towards the oral cavity so you can spit it out or swallow it. And if you swallow it, the acid in the stomach will kill whatever bacteria is trapped in the mucus. So that kind of is important. All of the chemical secretion and the layers that you have in the surface barrier first line defense mechanism. Now, these first line defense barriers are also prone to injury because they are the outermost layer that we have in our body. So in case if these surface barriers are breached, then you have your second line defense mechanism. So second line defense mechanism includes these five things: phagocytes, natural killer cells, inflammatory response, antimicrobial proteins, and fever. Phago means eating, phagocytes, the cells that eat. Natural killer cells also do the same thing. Inflammatory response, inflammation itself is geared towards containing the infection from spreading to other parts of the body. Antimicrobial protein like interferons and complement proteins, and then fever. Let's start with our second line cells or second line defense with phagocytic cells. The main one that we have are neutrophils. From the previous chapter, you know that we have a lot of neutrophils in our body, and that is because it's a phagocytic cells. It loves to eat up all the dead cells, bacterial cells, and make sure that it protects our body. The Other than neutrophils, we also have macrophages, which is more voracious than neutrophils. Macrophages are basically cells, monocytes that escape the blood circulation and goes into the tissue. In certain parts of the body or organs, these cells, macrophages, are permanent fixtures. Like you have it in liver, you have it in microglia, and those in your brain, those kind of things. Where fixed macrophages are free macrophages. They basically go in and out whenever they want, and they wander through the tissue looking for any damage or any kind of bacteria that might have breached the surface barriers. So these two are your phagocytic cells. The process of phagocytosis itself is important to note. In this picture, it actually shows you all the different types or steps of phagocytosis. The phago means eating. So the first part of step of phagocytosis is that it has to identify the antigen, that it does not belong to you. Once it identifies it, it basically puts forth the podocytes around it and forms a vesicle or phagosomes or phagocytic vesicle and takes it inside its cytoplasm. And then you have the lysozyme, which usually contains all kind of toxins, oxidizing agents like hydrogen peroxide. So that lysosome, with it full of its enzyme, merges with the phagosomes, and it kind of lyses the cell membrane and kills the bacteria. And whatever is left behind, it's just let out. It's important to note that whatever protein from the bacteria, these phagocytes also display on a special receptor on their cell membrane for other immune cells to identify it and be cautious about this antigen that it just digested. So phagocytosis or phagocytic cells are not only there to invade and kill all the antigens that might have entered our tissue, but it also helps to um, kind of like a flag of what it ate, so the other immune system will be aware of in case these bacteria are in other places as well. So the process itself is called as phagocytosis. In this page, you'll see all the free radicals, oxidizing chemicals, and pH, and all of those found within the lysosomal enzymes in your phagocytic cells. Natural killer cells are non-phagocytic cells, except they also kill antigenic cells through apoptosis. So basically over here, what they do is it identifies the antigen with help of a receptor, because they lack our own receptors that other cells have in our body. And the immune cells, since they do not identify those receptors as self receptors, they at once causes this viral infected cells or bacterial cell to undergo apoptosis, and then kill that cell. So you have your cancer cells and viral infected cells basically killed through natural killer cells like this. They also cause inflammations through potent chemicals that they release, which leads us to the next second line defense mechanism that we have, which is called as inflammation. Now, inflammation is usually viewed as a wrong or bad thing that happens in our body. Technically, inflammation basically tells you there's tissue damage and it has to be contained so it doesn't spread. So if you have inflammation because you kind of sprained your ankle, that means you have inflammation in that area because you have tissue damage there, and it actually helps in repair of that broken cartilage or ligaments or whatever it is faster. In the case of inflammation where you have toxins in, like in an insect bite, or if there is another kind of inflammation where there's a tissue damage that's causing inflammation. So it basically contains, due to bacteria, it contains those toxins within that area and prevents it from spreading to other portions, which actually also sets a stage for repair in that area. So benefits of inflammation is prevents the spreading, disposes the debris and pathogens, alerts the adaptive immune system, and also sets stage for repair. The worst part of the inflammation of it is when it actually hinders the function, so you can't move your joint or something like that. The pain-causing stimulus that you have because of the damaged tissue itself and the chemicals that are released by the damaged tissue, and also swelling. If the swelling happens in certain areas, say for example in your throat when you have anaphylactic shock, so that inflammation is systemic and it kind of obviously prevents you from breathing and you die. So that's why we don't like inflammation. Any kind of inflammation, there are four cardinal signs. You have redness, heat, swelling, and pain. Rubor, color, dolor, those kind of things. So there are four important things for an acute inflammation that we have to notice: redness, heat, swelling, and pain. So now, fifth one comes with the impairment of function. Now, what causes all of these cardinal signs? Um, Vasodilation of your blood vessels in that area. Now, why would you have vasodilation in that area that it got damaged or toxin is there? First off, because of the tissue damage or of the toxins, now you have histamine being released from the mast cells, or you have your eosinophils also over there. It releases histamine. Histamine is a potent inflammatory chemical, which it causes vasodilation in that area. Now, once you have vasodilation, the fluid from the blood starts to seep into the tissue, which causes edema in that region. And it also helps your macrophages to go through those porous, vasodilated capillaries into the cells so it can start repairing or eating away any bacteria or toxins that might be in the tissue of the damaged area. Um, because we have vasodilation, you have redness over there. Because of vasodilation, you have more blood flow, which leads to heat. Because of vasodilation, more fluid seeps into the tissue, causing edema. And because of the damaged tissue, chemicals release histamine and all of those, you have pain. And because of the swelling and pain, you have impaired function. So everything starts with that area. Okay. So again, inflammatory chemical release, vasodilation, increased vascular permeability, phagocyte mobilization. So these are basically the stages of inflammation that we have. The chemical released is once again, histamine released by mast cells usually, and it's an inflammatory chemical. And then the other ones you have kinin, prostaglandin, cytokines, and all of those. It doesn't matter which one, it causes vasodilation and locally causing the capillaries to become leaky, and that attracts phagocytes to that area. Phagocytes can easily seep out and eat up everything. So here's your vasodilation, increased permeability. It causes hyperemia, which is the redness, and allows the exudate, which causes the edema in that area, as well as pain because of edema. Vascular permeability leads to macrophages to go through. That basically helps in removal of damaged tissue or invading foreign antigens over there. The last one is phagocytic mobilization, which is basically how does the cells from the blood know exactly, like macrophages, know exactly where to get out of the capillary and go into the tissue, and how do they know once it's in the tissue which exact place is a toxin or antigen is located within the tissue. That basically are the stages that you have for your phagocyte mobilization. Okay, so you have a tissue damage because of the nail that you see over here, and now you have a release of bacteria into the tissue and also some chemicals that are released. And that itself causes vasodilation because of the histamine that's released over there. And that vasodilation basically leads to the first step, which is leukocytosis, which is increase in WBC in a normal situation to prevent any kind of diseases. So now you have neutrophils, usually responds first because they are your phagocytic cells. And then neutrophils basically go along the margin in the area where the damage is because it's porous. Now that's called as margination. And then through the pores, they squeeze out and go into the tissue called as diapedesis. Once they're in the tissue, the neutrophils, they go straight to the damaged area, not over here. The reason why they don't move somewhere else where there is no damage is because they go towards the trail of chemicals that are left behind by the bacteria or the damaged tissue. So the process where they go and find that scent is called as chemotaxis. They're following the chemical trail. So once they reach that area, they start doing their job, engulfing the bacteria, removing the dead tissue, removing the toxins, those kind of things. So these are the stages of phagocyte mobilization that we have. Um, once the macrophages or phagocytic cells eat up all the bacteria and everything, they actually become dead after a certain number of bacteria being engulfed. Dead neutrophils, dead tissue, and dead pathogens all together forms this creamy yellow mixture called as pus. Pus usually comes out of the body, and it should. If it doesn't and is within deep inside a tissue or an organ, it's called as abscess. The main difference is that these area is actually enclosed, this pussy area is enclosed by thick collagen fiber, and it is to wall off any kind of infection that might still be live bacteria that might still be within that pus. Now, it cannot stay in our body for a long time, so it has to be surgically drained and then clean that area and let it heal eventually. The problem with it is that certain bacteria, when gets engulfed by a phagocyte, they actually is not killed by the lysozymal enzymes. Instead, they actually thrive within the phagosome, and those bacteria, like tuberculosis bacilli, can cause diseases. So a person can have TB for many, many years and not have any symptoms. The reason why they do not have symptom is because once the macrophages know that they haven't been able to kill the bacteria after engulfing them, they wall themselves off to form a granuloma kind of thing. It is kind of like a connective tissue that wall themselves off and to prevent the bacteria from escaping the macrophages and also into the system. So that is very important. So that's why a person without can be without symptom and still be TB positive because they have the bacteria within the macrophages walled off, and so they don't have any symptoms. They don't get sick, but they test positive because they still have the bacteria, and your immune system has been alerted of the presence of the bacteria. The next second line defense that we're going to talk about is antimicrobial proteins. You have your complement proteins and you have your interferons in there. So they're able to directly attack the microorganism or hinder the ability of reproducing of the microorganism. Interferons are usually seen when you have viral infection. So these chemicals actually warn the neighboring cells that they have been attacked by a virus. So interferons does not help the cell that has been attacked by the virus, but they actually help the healthy cells around them that to prevent themselves from being attacked from the future virus that comes out of the sick cell. So there are different kind of interferons based on which type of cell produces it, and the way they work alerting the neighboring cells of a possible viral infection is this. The first one, as soon as the virus enters, they start replicating. So once they replicate, they go out killing the host cell. as soon as the virus enters the cell number one, the RNA is triggered. So the DNA starts basically becoming mRNA. mRNA becomes the interferons. These interferons actually go to the neighboring cell and warn them. And by warning them ahead, this host number two cell, neighboring cell, is able to produce an antiviral mRNA, which then becomes antiviral protein. So when you have new viruses coming out of the first cell trying to infect the second cell, it is already prepared with the antiviral protein, and which prevents the replication of the new virus in the second cell as well and replicating further. So this is basically how the interferon mechanism works in our body. Complement proteins actually have, there are 20 smaller blood proteins that are circulating in our blood. Your textbook talks in much detail about it, but I'm just going to point out the importance of the complement protein is that it alerts your adaptive defense system. It can also cause inflammation going on and wall off the infection. But the other important thing is these 20 proteins, they come together to form a complex called as membrane attack complex, which forms a pore within the cell that has to kill. And that pore basically opens the inside cell environment to the outside cell environment and causes a whole bunch of water to get in and causes lysis of the microbe itself. So you can see here's the pore that's been formed within the antigenic cell that needs to be destroyed. As soon as the pore, it's like a faucet opening. So it just whatever electrolytes imbalance happens and causes flooding of the water inside the cell and destroys the target cell. Next one, the last one that we have of the second line defense is the fever. Fever is usually not appreciated. We usually take anti-fever medication and all those stuff, but fever itself is to increase the toxins that we have in the blood due to an infection. When the macrophages are exposed to these things, macrophages starts to produce something called as pyrogen, which then goes and alerts our brain system to increase our thermostat, which then increases our body temperature. The reason why we have to have body temperature increased when you have infection is that it basically increases the metabolic rate and repair rate, and also sequesters zinc and iron needed for microorganisms for their replication process. So that basically is important. The reason why we have fever important. Now, I'm not talking about very high fever where you start denaturing your own protein. I'm talking about mild to moderate fever where you can actually decrease the multiplication of the pathogens in our body. So those are the second line defense that we talked about. Now we're moving on to the third line defense, which is adaptive immune system. Adaptive immune system is a very specific immune system against a specific pathogen. So if you have two type of bacteria, the adaptive immune system will have two different mechanisms to attack these two type of bacteria in our body. So one mechanism will not be like in the innate where it basically attacks pretty much anything that does not belong to you. That's not how adaptive works. Adaptive is more systemic, because you're dealing with B cells and T cells, which are in our blood. So wherever the blood goes, the immune system goes with it. And it takes a little bit of time to prime itself, to form that specific response attack against a particular pathogen. It needs to prime itself, analyze the bacteria or antigens, and then start attacking it in our body. So characteristics are specific, systemic, and it has memory. The memory cells basically protects us from taking too much time if the same bacteria attacks us twice. There is no need for us to do the priming again if you have one bacteria attacking our body twice. So to avoid that second priming, unnecessary second priming in our body for the same bacteria that we already have been attacked by, we have what we call as memory cells. So if we have memory cells, we know we can identify which bacteria we got attacked in the first place and then easily form a response without wasting any time. So that's very unique to your adaptive defense mechanism that we have. Now, the two branches depends on which cell we have. The humoral, which is the B cell, and the cellular, which is the T cell in our body. So we're like humoral means anything that is in the fluid, bodily fluid. So that's your blood, that's your saliva, that's your secretions of any kind, placenta, everything. Humoral is through B cells, which means B cells produce antibodies, and antibodies go into any fluid that we have in our body and basically spreads to those areas. And the importance of humoral is that because the antibodies are released into the fluid, these antibodies cannot know what is within the cells or tissues. So whatever is inside the cytoplasm, intracellular target like viruses or cancer cells or transplanted cells. So the B cells cannot, or the antibodies cannot recognize whatever's hiding behind the cell membrane or inside the cells. So humoral immunity is considered an extracellular target. So anything that's free floating, toxins in the blood, any bacteria that's in the blood, or any kind of mismatched blood, so it binds to the RBC that does not belong to you and form agglutination. Those kind of things are what done by humoral immunity. Cellular immunity is your T cells, which actually recognizes what is behind the cell membrane. So has it been affected by virus, or is there a mutation like for cancer cells? Or does that cell even belong to you like in transplanted cells? So that's basically what your cellular immunity identifies. The lymphocytes, the T lymphocytes in your cellular immunity, there are two mechanisms. You have direct and indirect mechanism. Direct is killing the infected cells directly. Indirectly is through inflammatory response, macrophages, and those kind of things. The next thing is the antigens. Definition of antigens, antigenic determinants. So antigens are any substances that basically provoke immune response in our body and mobilizes adaptive defense. So anything can be an antigen. Um, the classification of antigens are complete antigens or incomplete antigens. And in an antigen, not the entire cell or tissue is responsible for provoking an immune response. It's only a part of it, and that particular part of it that provokes the immune response is called as antigenic determinants. Antigens can be self-antigens, which means your own antigens. These self-antigens are basically receptors that are found on each of our cell, which are identified by our WBCs as self-antigen, and they do not harm it. So they do not and should not harm your own cells. That's why it's called a self-antigens, your own thing. In case the immune cells fail to respond self-antigens, that's when you have your autoimmune diseases. Okay. A complete antigen has two properties, immunogenicity and reactivity. Immunogenicity, proliferation of specific lymphocytes, reactivity, able to react with the lymphocytes that was activated or the antibodies and cause an immune reaction. So these two things are important for you to be a complete antigen. An incomplete antigen is by itself, they do not provoke an immune response, but when they go into the body, they bind to the proteins in our own body. And that complex that forms with the hapten self protein that we have in a system now, that complex causes an immune response in our body. That's why it's called as incomplete antigens or haptens. It can be poison ivy, dander, detergent, cosmetics. So all of our cosmetics are nanotechnology, and the nanotechnology in our body has not been researched heavily. So Cosmetics with nanotechnology is bad because they go inside a body and we don't know what these chemicals does to our immune system yet. The same goes for detergents as well. All the smelly things that we add to our clothes when we wash it, those smelly things are basically haptens, which can cause allergic reaction. So, spraying like Spring Fresh spray into your room or in your clothes, those are all spray that lingers in our body. You inhale it and it goes into your mucus through the mucus system and it becomes a hapten and elicit an immune response in your body. So it applies for poison ivy and dander as well. So, antigenic determinant. This is what I said when I talked about antigenic determinant. The entire antigen does not cause an immune response. It's only portion of the antigen that causes immune response. So if you look at this area, and this area. So these are where your antigenic determinants are. And there are specific antigens that bind to specific antigenic determinants. In this case, you're looking at one antigen with three different antigenic determinants, which usually you don't see like this. Usually have the same one. But if you have different antigens with different antigenic determinants, then you'll have different antibodies that are able to bind to these different antigenic determinants of two different bacteria or antigens. The antigenic determinant or antibodies are created to fit with this particular antigenic determinant. So it's like a lock and key mechanism. You can have only one key for a lock. Okay, so you can't have multiple. Pretty much like that, you can only have one antibody binding to one antigenic determinants at a given time if everything works fine. Self-antigens are the receptors that are found on our protein or cell membrane. These are your surface receptors or surface proteins that we have in our body. So pretty much all cells, when they're formed, usually have these surface receptors or self-antigen on them. We also call them as MHC protein. There are one set of important self-proteins or surface receptors called as MHC proteins, major histocompatibility complex. Now, these MHC is very unique for each individual of us. The closer these MHC complex matches from one person to the other person, the better the chance of transplanting organ between these two people. The further apart the MHC complex is, the more issues you're going to create with the immune system when you do a transplant in them. So these MHC proteins are very, very unique surface proteins that you have, and they basically help us to identify our self-antigens, or these are your self-antigens. So basically, they help us to our self cells to stand out from the foreign antigens if they invade our body. Um, you have different type of cells that are very important, and when it comes to adaptive immune system, you have what we call as your T and B lymphocytes, and you also have what we call as antigenic presenting cells. These antigenic presenting cells are your B cells, you have your dendritic cells, those kind of cells which actually engulfing neutrophils, macrophages, they engulf it and then they display it. So those are antigen presenting. So after engulfing, whatever pieces there are of the antigen, it gets presented. Hence, antigenic presenting cells, APC cells that we have in our body, we'll deal with them eventually. Now, when we come with the lymphocytes, with both B and T lymphocytes, it's both of them are formed in the red bone marrow of our body. But after forming, after the origin, what happens to those lymphocytes differs. When it goes to maturation, the T cells basically go to the thymus to get matured, and then the B cell remain in the bone marrow to become mature. So maturation process depends on which cell we're talking about. Is it going to thymus? That'll be your T. If it stays in bone marrow, it'll be your B lymphocyte, B for bone marrow. Once that is done, the maturation is done. in the either the thymus or the red bone marrow. Now you go on to the lymphoid system, lymphoid bodies, secondary lymphoid organs. In these secondary lymphoid organs, now remember, lymph itself takes out fluid between the interstitial space and then in the interstitial space, and then takes all that fluid and dumps it back into the venous system. Now, when they're taking out the fluid, there's a high chance that it might be any kind of breach in the surface barrier, which leads the bacteria to get in, which means lymph nodes will have all kind of bacteria in it. So when you have these immature cells from the thymus and bone marrow going into your secondary lymphoid organs like your lymph nodes, they're getting familiarized with all the antigens that are possible to get inside our body to cause disease. So they are basically um, introduction course of all the different types of antigens that might encounter, that those cells might encounter later on in their lifespan. So that is very important, where they get secondary seeding, and then they get antigen encounter and activation. Once they get encountered by the antigen, they have to get activated. So the activation is by replication process, and that's basically what activation means. So once the cells bind to this antigens in the lymph node that has been blocked or neutralized, these cells basically now are ready for activation, so multiplication. So they basically, if it is a B cell, they start to form same kind of antibodies that can bind to these antigenic determinants. If it is T cells, they're going to be getting ready for engulfing the or killing the antigen in there. Proliferation and differentiation. It's how do these cells multiply once they're activated with the help of the antigenic determinants from the antigenic cells. It's through what we call as cloning process. Cloning is basically where they have identical DNA or genes. So once you have identified the antigenic determinants, you don't want to change it when you're doing a cell division. So that's what happens usually during a cell division, where they kind of keep changing. But you don't want the cell to change because you've already identified the antigenic determinants. All you want is to produce more of the same type of cells that can identify the antigenic determinants. So that's why you have cloning process, where you have the identical cells copy of it, where it can resemble or identify this antigenic determinants that entered our body. That's what cloning is. The cloning process ends in two type of cells. One is the effector cells that fight off the infection, and one is the memory cells for future infection of the same kind of bacteria that you might have. So that is your last step, proliferation and a differentiation process. Now, the other thing we have to think about is you have millions and billions of bacteria and viruses out there. So how does your 25,000 genome that you have is able to produce so many different type of antibodies that can bind to so many different types of antigenic determinants on an antigen? It's through a process called a somatic shuffling, where you have a set of genes responsible for forming antibodies, and that set of gene gets reshuffled again and again until the perfect pair is found, which fits perfectly to your antigenic determinant. Now, I'm not talking about the DNA binding to the antigenic determinant, but I'm talking about the mRNA that is formed from that perfect DNA sequence that gets shuffled back and forth, and that basically forms your antibody, which is capable of binding to that particular antigenic determinants that you have in the body. So that's your somatic shuffling that you have. Once the lymphocytes are formed, education during maturation, how do you know that you have the perfect type of lymphocytes that have been multiplied? It's through positive and negative selections that we have in our process. Positive selection in this picture shows you exactly how if you fail to recognize the self-antigens, then you'll be killed off. Positive selection. So you have to positively identify the self-antigen that's on the cell. Okay. The second step is negative selection, where you should not bind to the self-antigen, the MHC, on that self receptor that we have. So the self-antigen, that's your MHC, that carries the protein either from within your own cells or the cell that it has engulfed through APC cells. So whatever it's displaying out, whatever piece of protein the MHC is displaying, should not, if it is your own cell, should not attack it. Okay, so if they bind to it, then you're done. Like in this case, it's tightly bound, which it has recognized self-antigen, which means it'll cause an immune reaction onto your own body. That's why you'll have an autoimmune disease. So that's why this cell, where it recognizes the self-antigen and it'll start attacking it, will be killed. Only the cells that recognize the receptor but that does not bind to the self-antigen will pass. Okay? So self-antigen should not be attacked because it's yourself. So those are the cells that goes into maturation process. So this is how your T cells are educated in your thymus. B cells is more like antibodies are released, it goes binds to the antigenic determinants, and that's it. So those process involves more into cloning, which we're going to talk about eventually. Antigenic presenting cells, APC cells, are of dendritic cells, macrophages, and B cells. These guys basically engulf or kill whatever antigen it's out there, and they start displaying on themselves for your other adaptive immune system like T cells and B cells to recognize, and then helps in production of antigen and killing of the cell itself. Dendritic cells, we talked about when we were talking about the skin in 226. They're just wandering around, phagocytic cells. So when they wander around, they can eat up a bacteria. And once they eat up, they present this bacterial protein that was just digested by them to the adaptive immune system. Macrophages does the same thing. They're phagocytic cells as well, and they also cause inflammation in the body. B lymphocytes basically do not activate naive T cells, but they help in assisting the T cells. We're going to start off with humoral immune system first in detail and then move on to cellular immune system. Humoral immune system is by B cell that we have, which means it produces antibodies. This is where you need to have a specific B cell producing a specific antibody that attaches to a specific antigenic determinant in the antigen itself. So that's where you have your clonal selection made. So a certain type of gene combination forms a perfect antibody to fit to a perfect to the antigenic determinants. So you want only that type of gene combination. That's where you have your clonal selection. So what you're seeing over here, the primary and secondary response. Primary response is when a body is exposed to antigen for the first time. So what we're seeing with COVID, because it's being a novel virus, new virus, we're seeing primary response in all of the people after they get infected. So primary response is you have the antigen in our body, and the B cells, there are different combinations, and it's kind of rambling around finding out a perfect fit for that antigenic determinant. Once it finds the perfect fit, perfect antibody for that antigenic determinant, it starts activating the B cells for proliferation, for cloning process. So that's what you're looking at over here. It forms the clone. And these cells become the effector cells, which is the plasma cells, which produce a whole bunch of antibody and a memory cell that is there. Now, when you have the same infection second time, and that's basically where you already have the memory cell made, so you don't have to go through the clone process for the secondary response. Once the second time the same antigen is being exposed, you already have the memory cells, so they at once go and form a whole bunch of plasma cells and a whole bunch of antibody and also a memory cell there. Now, this is what basically the problem in the COVID situation, because they're saying these antibodies that are created are only lasting for 3 to 6 months. So there's a chance of reinfection of COVID in the same person after 3 to 6 months with the COVID. And keep in mind, COVID being virus, it mutates constantly. So let's get back over here. So in this graph, you're looking at the titer of the antibodies that are produced in the primary response and secondary response. The primary response, because they have to have priming, you have about few day delay in here before the antibody starts to produce. Okay, that delay might be dangerous for certain type of infection, so you have to intervene and do something about it. So even after the antibody is starting to produce, you'll see the titer is not high enough. It's very low. But if you give the second exposure, the same antibody, same antigen A, sorry, not antibody, antigen A for the second time, you'll notice there is no spot priming. You give it on the 28th day and you have a spike of antibodies on the same day because you already have memory cells formed. So you don't need to prime over there. You have plasma cells and a whole bunch of antibody, and the titer is extremely high compared to the first time. Now, if you give a second antigen, okay, first exposure, second antigen B, this response for the second antigen B will mimic the first antigen, and titer will be less because you still need the priming done for the second antigen as well. Okay. The different types of humoral immunity depends whether it is active or passive. So the active one is one where you have memory cells made. The passive one, you do not have memory cells made, which means you can get reinfected again. So naturally acquired active, artificially acquired active. Naturally acquired is where you get sick, you form your memory cells, and you basically, when the second exposure, you are ready. Artificially acquired is where you put vaccine of an attenuated antigen or a dead antigen into our body through a vaccine. You get exposed to it, your body primes it and forms the antibody. The first exposure is basically ready. The memory cells are formed. So when the actual virus comes in or bacteria is encountered in your body does not require that few days of priming because it's already done through vaccination process. So the response in our body will be immediate with high loads of antibodies, high titer of antibodies, so you can react to it appropriately, fast, and heal or cure yourself. Passive, on the other hand, I said there is no memory cells formed. So it can happen naturally through placenta, through milk, from the mother to the fetus. That's naturally acquired passive humoral immunity. The artificially acquired is that where you get the disease or toxins are in your body and you do not have time to prime the body because during that priming moment you will lose the patient. So in order for you to protect the patient, you give ready-made antibodies to that patient and save the patient's life, like in snake venom, where you give the antibodies that binds to the toxin of the snake venom and then helps to cure the patient. Or botulism, where the botulism toxin is too much and causes paralyzing muscles. So you give antibodies, ready-made gamma globulin, to prevent it from respiratory paralysis and death. So these are the different types of humoral immunity based on whether or not you have memory cells made in the body. Now I keep talking about antibodies. Antibodies. What are antibodies? Antibodies are immunoglobulin, gamma globulin. Basically, you can call or antibodies. They're all the same. They're freely floating proteins in the blood, and they bind if the antigenic determinants fit into these antibodies only. There are 5 classes of antibodies. We call it as MAG, IgM, IgA, IgD, IgG, IgE. So you have 5 classes that we have, and each one is different in structural and their function as well. But each of these different classes of antibodies have a basic structure. They are basically made of four chains of polypeptide. So you have two identical heavy chains and two identical light chains. And then you have variable region at the end of each arm to form your antigen binding sites. You also have a stem which forms the constant region. Um, so then yeah, let's look at the constant region is the determinant's class of antibody in the constant region. So you have here's your heavy chain, light chain, antigenic binding site that is usually on the end and either side right there. You have the heavy chain and light chain over here. So you can see all the different types of polypeptide chains that come together to form these antibody structure, which is usually white type structure. Let's talk about IgM. IgM is a pentamer, is known for used for agglutination, and it kind of the first antibody that is released in your body. So IgM is the first antibody released. So in the COVID case, if you get IgM, that means you've been exposed recently to your COVID, and then follows is the IgG, which is the secondary and late primary response that you usually have, and it's a monomer. So I would strongly urge you to go over this table where it tells you exactly where these type of antibodies are used in our body. So because it is a pentamer, the IgM, it can actually bind to different antibodies in each of these um, binding sites. This is what causes the coagulation of your mismatched RBC. So it kind of forms kind of a link, and it binds to multiple RBCs, the donor RBCs that was mismatched initially, and causes agglutination. So it's the first primary response that you have, first immunoglobulin that responds in your body. Then you have IgA. IgA, it's a dimer. You have your disulfide bond in between them, and it is released into secretion. So you'll see them in saliva, sweat, milk, intestinal juices. So you'll see IgA even in your fecal matter. In your urine. So those kind of secretions that you have, technically not in your urine, but in your fecal matter, you'll see that. So this IgA is important in in the first milk that is given by the mother to your newborn baby. So this IgA is the first immune system protection that the newborn gets from their mother through their milk. So that is your IgA. Then you have your IgD, which is also which is a monomer that you have, and it actually is found in the cell membrane of your B cells. So it forms a receptor of your B cells. And then IgG, again, like I said, it's the late primary response or secondary primary response. So it's a main antibody that you have, protects against bacteria, viruses, and all those things. And it also crosses the placental barrier from mother to the fetus. So 75 to 85% its circulating antibody is your IgG. Then IgE is it's usually not that much found it because it causes immune reaction, inflammation in your body. It's just like eosinophils that causes your histamine release, pretty much like that. IgE also binds to mast cells and basophils, and they basically cause inflammation and allergic reactions in your body as well. And it crosses your secreted through your GI tract, respiratory tracts, tonsils, and all of those things as well. Again, it's allergic reaction that you have, IgG. So go over all of these antibodies, their different classes, and their function of it, and their structure of it as well. Um, so once they bind to these antigens, these targets, what happens to these antigen antibody complexes? So you can actually have certain things happen after you have an antigen antibody complex. You can neutralize, you can agglutinate, you can precipitate, or you can have complement fixation done. So let's look at this a picture that we have. So this is what happens after you have an antigen antibody complex formed. Neutralization. So the toxins that are freely floating, the antibodies bind to those toxins and make it less toxic or non-toxic. So it can be exotoxins of bacteria or viruses that can bind and makes it less effective in our body, and so hence diseases doesn't happen. Neutralization. The next one is agglutination. Like I said, your pentamer basically binds to each of these antigen binding sites, bind to the RBCs and agglutinates that. After that is the next one is precipitation. So these are tiny soluble antigens in the blood, so they kind of bind it and make it more prominent either for macrophages to kill it or destroy it, whatever. Complement activation. So what they do is they go and bind to an antigenic cell and they alert it. It's like a red flag on the cell membrane of the antigenic cells. So the complement protein identifies these red flags, these antibodies that are bound to the cell surface of the antigens, and then they form the MAC, the membrane attack complex in that cell membrane of the antigen, and which leads to lysis. Now, what happens to all of these after neutralizing, agglutinating, and precipitating? What happens to the end product after it does that? It ends up getting phagocytosis or inflammation and contained or cell lysis themselves. So, depending on what is happening, either neutralized, agglutinated, or precipitated, or membrane attack complex, you have cell lysis, inflammation, or phagocytosis done. So technically, you have what we call as plan: precipitation, lysis, agglutination, neutralization. So this is basically what happens to your antigen antibody complex after it is formed. Now, when you have antigens that are too large for it to either precipitate or lyse or agglutinate or neutralize, what happens is the IgE gets triggered. IgE basically starts to destroy the surface of the worm, and it causes lysing from outside to inside of your worm cell membrane or the skin of it. Um, Antibodies are very much used in a lot of purpose of research, um, and we need a specific type of antibodies formed. So when you hear about the immunotherapy that is out there for different type of cancers, you're talking about these lab-made antibodies. We call it as monoclonal antibodies. So what happens is these you have a receptor on an antigen. You know the formation or the sequence of those amino acids on the receptors in the antigen. Now, with that help of that, you can actually form. It's like a lock and key mechanism, right? Once you have the key or the lock, you can easily make a key for that particular lock. Pretty much like that. Once you have an antigen receptor, which is the lock, you can form a key, which is the antibodies in a lab setting. It's kind of what fits perfectly. It's like a mold. The opposite of that mold, you have the perfect antibody. How do you make these specific antibodies in large quantities in a lab setting? Once you identify which gene combination produces the antibody that fits perfectly to that particular antigenic receptor, what you do is you take that gene, put it in a hybridoma, or we call it as a cancer cell, B cancer cell, and then form a hybridoma, which is a tumor cell that can produce the same kind of antibodies in a large quantity. because the cells die after 50 replications normally, you don't want them to die. Hence, you're fusing it with the cancer cells so they can reproduce. And every time they reproduce, you have more and more antibody produced in large quantity. So once you have that antibody produced and released out of the cell, you can collect all that and give it to the patient. So that is what immunotherapy is all about. So once you know a specific receptor of that specific cancer, you have the lock. You can make the key in the lab. Once you make that key, you take that, merge it with the cancer cells. Now you have the hybridoma. You can make large quantity. And once you have the antibody removed from the lab setting, you can give it to the patient. Technically, theoretically, the antibodies given from the lab to the patient should go and attach to the receptor on the cancer cells and kill the cancer cells. Or you can radiate it, and once they're flagged, you can pinpoint and radiate and have a localized radiation given to that particular person for that particular cancer. So the first thing they usually do in cancer treatment is isolate the cancer tissue and look for the genetic combination of that cancer, and then see if they can have immunotherapy given radiation or chemo, what is the best way to attack it. In the clinical setting, in the research setting, antibodies are used in many testing things like HIV testing, today COVID testing. So there are a lot of testing that can bind to that specific area of the antigen and it will flag. And if it flags, the color changes and it's positive or negative. So once again, antigen antibody complex is there. They do not destroy, they prepare them for destruction. That's what plan is all about. And then the only type of antigen these antibodies can attack are extracellular that are in the blood. So they don't actually go find what's hiding within the cells. For that, you have your T cells, which will be the next portion of the lecture. So cellular immunoresponse. This is your T cells directly attacking and killing the cells that do not belong to your body. So what cells are they? Viral infected, bacterial infected, cancerous, abnormal, or transplanted cells. These are the cells that your T cells will attack. So they are intracellular pathogens or antigens, except they're intracellular antigens. T cells, intracellular antigen. B cells, extracellular antigens. There are different kind of T cells that we have in our body. There are CD4s and CD8s. CD4s becomes helper T cells and some regulatory T cells. CD8 becomes cytotoxic T cells. Now, what are these CD4 and CD8? They are the receptors that are found in certain type of T cells. So if the T cells have CD4 receptors, you call them CD4 cells. And these CD4 naive cells, they end up becoming mature helper T cells or regulatory T cells. Helper T cells basically activates your immune system. So without helper T cells, you have zero adaptive immune system. Now I'm saying adaptive, not just cellular, because helper T cells also initiates your B cells, your humoral immunity as well. Regulatory T cells, it dampens the immune system, preventing autoimmune reactions in your body. So regulatory T cells kind of make sure the T cells are not overreacting in our body. Your CD8 cells becomes your cytotoxic T cells, which are your killer cells. So these are the ones that kill your antigens which are hiding behind the cell membrane. So that'll be your foreign transplanted cells or mutated cells, those kind of cells that we have. Um, the way that these cells get activated, whether CD4s into helper T cells or CD8 into your cytotoxic T cells, is that you require your MHC cells. There are two kind of MHC proteins that we have on our cells. One is class 1 and the other one is class 2 MHC proteins. Class 1 MHC are found in pretty much all of your body cells except for RBCs. Class 2 are only found in antigen presenting cells. Those are your dendritic cells, phagocytic cells, and B cells. So these class 1 and 2 MHC proteins, if you look over here, you have your class 2, you have your class 1. Class 1 is found in all cells except RBC. Class 2 is found in antigen presenting cells. Okay, that's what we said, right? So let's look at what is the main difference and importance of MHC class 1 and class 2. Now, your T cells, as they are maturing, so you're looking at here bone marrow, lymphocytes are formed, they're immature, they go to the thymus for maturation process. In your thymus, you have two type they divide into. One that has CD4 receptor, one that has CD8 receptor. In order for these CD4 and CD8 to become either helper T cells or cytotoxic T cells, you require class 1 MHC or class 2 MHC proteins. Now, class 1 MHC is found in all cells except RBC. Once they here's the antibody or antigen on class 1 MHC protein from all of your cells. They display to CD8 and they become cytotoxic T cells. Cytotoxic T cells kills a cell that is either mutated, cancerous, or viral infected, those kind of things, or transplanted, right? So all cells are capable of producing cytotoxic T cells. All cells can get mutated and become cancerous or infected by virus. So they have to be killed by cytotoxic T cells. That's why class 1 activates CD8 cells. Now, if you look at this side, class 2, over here, it's activated by APC, antigen presenting cells, which becomes helper cells or regulatory cells. Now, antigen presenting cells are your phagocytic cells, dendritic cells, and B cells. So helper T cell, specifically helper T cell, helps to basically activate your adaptive immune system, including B cell. Antigen presenting cell, one of it is B cell. So CD4s are activated by class 2 MHC protein to become either helper T cells or regulatory T cells. Regulatory T cells dampens your immune system in general. That includes B cell as well. So that is very important things that you have. The antigen presenting cells engulf phagocytic cells, right? So macrophages, dendritic cells. So those cells take antigen from outside and display it for your CD4 to get activated into helper T cells and regulatory T cells. Whereas class 1 is endogenous protein. This is exogenous. They have to take it from outside. This one is endo. They basically take a little bit of protein that they form within the cell and display it on the class 1 MHC protein for the CD8s to mature into cytotoxic. Endo, if your cancerous cells is there, the faulty protein from the cancerous cells is displayed out in class 1 MHC and then activates the CD8 so the cytotoxic can kill the cancerous cells. Endo, within, endogenous protein. Exo, outside, engulf, macrophage, dendritic cells. They eat from outside, display on class 2 for CD4s to become helper T cells and alert the immune system, including your B cells. Okay, so those are the important differences between class 1 and class 2 MHC proteins. You have a detailed differentiation of each of these class 1 and class 2. There you have, starting from where are they found, displayed, all nucleated cells except RBC, right? Class 2, it's found in APC only, recognized by CD8 over here, CD4 over there. Foreign antigens that are endogenous for class 1, exogenous for class 2. Cells displaying foreign antigens, MHC send this message. What kind of message does the protein, this endogenous protein, send to the cells? When you have an endogenous protein on class 1 MHC protein, it's saying, hey, this is what protein that I am making. Make sure it is not faulty, and if it is faulty, destroy me. Okay, so that is basically what it is showing, right? This protein, if it belongs to me, leave me alone. If it doesn't belong to me, then you can kill me. Okay, but class 2 MHC protein, it's exogenous from outside. It's saying, hey, this is what I ate. So this is the one that's lurking in our body. So be prepared. So that's why helper T cells and regulatory T cells are there. Okay, I captured this. Does not belong to me. This is a foreign one that I ate. So here's the thing that you have to be protected against. Make sure you are alert. That's what class 2 MHC protein tells you to your body, to your defense mechanism. Hopefully I was able to help you. And the other important thing of these MHC protein is the closest MHC proteins that we have to a transplanted cell, the better the acceptance of your transplanted organ is. The further apart the MHC it is to the donor organ, the more the immune reaction and the rejection you're going to have in the transplanted organ. So they're looking for the MH3 proteins on the cells of the donor organs. How close is it to your own MHC protein? The closer it is, the more acceptance the immune system will not react to it. The further apart it is, the easier the immune system will identify it as non-self antigen, and it'll start attacking and rejecting your donor organ. Now, activation and differentiation of T cells is, in order for T cells to be activated, you need two-step process. It's antigen binding and co-stimulation. So I'm going to be really brief on this. Your textbook goes into too much detail. So first off, antigen that has been presented by your class 2 MHC protein, APC, has to be recognized and as non-self antigen. So after you have the and dendritic cells engulfing and breaking down the protein and displaying on class 2, the CD4 cells recognizes that, and then at the same time you have something called as co-stimulatory molecule that also binds with your CD4 T cells over there. So it is not enough for the T cells to bind to the MHC class 2 and recognize the antigen that is presenting as non-self, but you also have to have the co-stimulatory protein binding to it. Why do you need both of them binding? Because apparently if T cells just bind to the MHC and the antigens that are presented on it, it might get used to it and it becomes inert. It doesn't react. You need that co-stimulatory molecule to put it in. When you start a car, you put it in gear and then you start and then put the gear up. So you basically need both of it to do in order for you to move the car. So that's basically how you have the T cell activation as well. You need not only the binding of the MHC class 2 and the non-self antigen, but you also need the co-stimulatory molecule binding over there with the receptor in order for the clone formation, in order for them to multiply and then eventually attack. Otherwise, they became inert and they do not respond properly. For it to multiply, your T cells require certain chemicals. We call it as cytokines. Interleukins are called as cytokines. So these cytokines help in cell development differentiation process in your immune system. So there are different kind of cytokines for different types of cells that we have in our body. You can just briefly go over it. Moving on to different type of T cells and their roles. Helper T cells, once again, without helper T cells, you have zero adaptive immune system. So zero immunity without TH or helper T cells. Your HIV attacks all of your helper T cells and kills them. So helper T cells are your CD4. Naive CD4 becomes helper T cells. So HIV binds to all of your CD4 cells, kills them all, so you end up with no helper T cells formed eventually, which is what leads to AIDS. So it helps to prime not only your cellular immune system, but also your humoral immune system. So it is both for T and B lymphocytes. So you can just go over all of this later. I'm not going to go into detail because they're going into too much details over there. The other type of cells that we have is your cytotoxic T cells. This is a killer cell. So they basically go and attack the cells that does not belong to you, or that has virus hidden or mutated cells. So viral infected cells, bacterial hiding in it, or cancer cells, or transplanted cells, whatever it is, cytotoxic T cells kills them. So how do they kill them? They produce two type of things, perforins and granzymes. The perforins forms these little pores on the cells that needs to be destroyed. So you can see the perforin pore right here. And through these perforin pore, the granzymes creep inside the cell that needs to be destroyed. These granzymes basically go into the nucleus and trigger apoptosis in that cell that has to die, and causes the cell death of that particular target cell. So that's how cytotoxic T cells kills them. Um, the next is regulatory T cells. The regulatory T cells are ones that dampens the immune system and prevents autoimmune reactions in your body. These are the cells that are being triggered or researched more when it comes to transplantation of a donor organ. So if you have regulatory T cells activated into a little bit higher than normal, then you can actually allow the donor organ to be accepted more readily, because these guys dampen the immune system. So there's less chance of rejection of the donor organ. Um, so this is what I was talking about, organ transplant and rejection of it. Last portion of the chapter talks about all kind of diseases in the immune system, starting with immunodeficiency. Immunodeficiency, you can be congenital or acquired. Congenital is your SCID, which is severe combined immunodeficiency syndrome, where the baby is born without any adaptive immune system, so there's no B or T cells. In this case, you have to have bone marrow transplant. You don't have to worry about donor matching because these guys do not have B or T cells, so there's less chance of rejection over there. Acquired is your Hodgkin's disease due to virus that is caused. So you have cancerous B cells that can lead to immunodeficiency. Hodgkin's is actually genetic mutation that happens in B cells causing them that disease. Acquired is HIV, where you have the virus that cripples your helper T cell. CD4 cells, remember, is attacked by your HIV. So when you're giving medication to an HIV patient, you're making sure that the levels of CD4s are high. If the levels of CD4 goes low, HIV virus goes high. That's what it means. If the levels of HIV virus goes high, CD4s goes even lower and lower. That means you have zero immunity in your body, which can lead to secondary opportunistic infections like pneumocystitis, pneumonia, Kaposi sarcoma, those kind of things, which is basically AIDS at that point. So you can be HIV positive, but not AIDS. HIV positive means you have the virus but under control. So you have enough CD4s in your cells and other immune system are functioning well. But if you have AIDS, CD4s are less, very less, leading to opportunistic infection. So people who have AIDS basically die of these secondary or opportunistic infections in them, not of HIV, but they die of other causes in their body due to less CD4 count in our body. We all know how HIV is transmitted. Unfortunately, apparently the stigma is gone because people are living longer with the HIV drugs, so they're being less careful. So you're seeing a rise in HIV, usually in younger people in the population. So HIV again is transmitted through bodily fluid that includes blood transfusion, contaminated needles, sexual intercourse, oral sex, mother to fetus, those kind of things. And then it's at once destroy this helper T cells, the CD4 cells, depressing the cellular immunity. So HIV can remain dormant in a lot of organs like lymph nodes. It can live integrated within our DNA in our dendritic cells that you see. So when you have these kind of HIV hiding within our DNA and not actually replicating actively and killing CD4, that period of asymptomatic period can last for years, and it can be there. Even with lower level of HIV virus in a patient, undetected load, we call them. Even in those kind of patients, you don't eradicate HIV viruses, you cannot kill them, you cannot cure them. They basically live within our cells. Like, for example, shingles, that basically chickenpox comes after years when we become old and our immune system starts to drop. That's because the virus that we got as a chickenpox is still in our body, in our nerves, that eventually causes shingles when we get old. Pretty much like that, HIV also can remain dormant within certain cells for longer period of time asymptomatic. So if you have stopped taking the HIV drugs, or if you have stressed too much in your life, then these HIV comes out of their dormant stage, and then you have now symptoms. Then you have to change the dosage, or you have to change the cocktail of the drug given for that particular for the HIV. Um, now, virus, we initially thought HIV only invades CD4, but now we're thinking that it also goes into brain and invades brain tissue and causes dementia as well. So there are a lot of research done in HIV and brain tissue, and see how they actually affect our nervous tissue as well. We're just talking over here about HIV and how the cell enters and all those things. The drugs that are given for HIV treatment are um, uh, targeting the replication process, the entering of the HIV into the target cell, and how they replicate within the CD4 cells, and how they basically form these new viruses within the cells and then go out. So you have entry blocking drugs, so they prevent the HIV from entering the cell itself. You have reverse transcriptase, because HIV is an RNA which becomes DNA once it enters our CD4 cells. It has to go backwards, RNA to DNA. We have DNA to RNA usually. Because they're going backwards, you have a special enzyme called as reverse transcriptase. So these drugs block that and prevent the virus from becoming a DNA, hence replicating within our cells. So it blocks that. It blocks the capsid that it forms after it replicates. It has to form a protein capsid around it before it can leave the target cell. So it blocks that capsid forming things from forming those things. So there are different drugs focusing on different parts of HIV replication when given. So usually give them as a cocktail. So you don't give one particular drug. You don't just give entry blocking drug. You give them reverse transcriptase entry blocking, as well as blocking the capsid forming drugs. All of them in a cocktail is given for treating an HIV patient. There is no cure. Keep that in mind. AIDS. There's no cure for AIDS. Okay, so you have to make sure that you keep it lower count, and make sure the CD4 is high by taking HIV drugs properly, stress, and eating healthy. The other spectrum of immune diseases are autoimmune diseases. Autoimmune diseases is basically where your own body attacks your immune cells attacks your own body. So they fail to recognize self antigens. Okay, so that's what is happening. Now, why do they fail to recognize your own or organ, own tissue? There are multiple reasons why they fail. There are different scenarios where you have autoimmune diseases. One scenario is they're saying that a cell might have gotten mutated all of a sudden, so now your immune system does not recognize it, or a barrier has been breached. Now you have a new cell that's exposed to your immune system, or you get infected by a cell, your antibodies are produced, your immune system is activated, except that antigenic determinant of that disease is so closely resembling your own self antigen that your immune system failed to distinguish between self and non self antigen. So they're so close together that they start attacking both. So that is what you end up with your autoimmune diseases. Okay. So failure of self-tolerance is the one that leads to autoimmune disease. Again, foreign antigens that resemble self-antigen, new self-antigens due to gene mutation, barrier that have been broken, hapten that has been attached. Those are the reasons why you end up with autoimmune disease. Now, when you have autoimmune disease, you start destroying your own tissue. So these are all the list of autoimmune diseases, few of them that we have in our body. It's starting to destroy your own cells. So, how do you treat autoimmune diseases? Because your immune system is the one that is attacking it, you suppress the immune system. So, that's the main form of treatment that we have so far for autoimmune disease. Suppress your immune system. Okay, so anti-inflammatory drugs like corticosteroids, blocking cytokine action so you don't multiply your immune cells, blocking your co-stimulatory molecules so you don't again cause replication and activation process. So these are all the treatment that we have. Ultimately suppressing your entire immune system. The problem with this, suppressing your entire immune system, you have secondary infections that you have possible. So once you see all those secondary infection, you reduce this dosage of the suppressing agents, suppressing drugs, immune suppressing drugs. That's basically how you treat autoimmune diseases. Hypersensitivity is you're reacting too much, right? That it becomes a threat to the tissue damage. So there are different types of hypersensitivity based on the how long it reacts and based on whether it's antibody or T cells involved in it. Antibodies are usually fast because they're free floating. So immediate and subacute hypersensitivities are antibody caused. Later one, delayed hypersensitivities are your T cells that help in it. Immediate are your type 1 and type 2. So you have your acute hypersensitivity, where it begins within seconds after the contact of allergens. So that'll be your food, any kind of IgE binding to the antigen, causing mast cells release, histamine release, and inflammatory chemicals, so asthma, those kind of things are all because of immediate hypersensitivity. Now, if you have systemic hypersensitivity, immediate hypersensitivity, it leads to anaphylactic shock, which is absolutely dangerous for health because inflammation is what? Vasodilation. And if you have systemic vasodilation, blood pressure drops. And also you have edema, which is what you have. You can't breathe when you have anaphylactic shock. So making it difficult for that as well. And vasodilation, blood pressure drops, organs are not getting enough blood, so you now have organ shutdown going. So you give epinephrine. Epinephrine, vasoconstriction, increases your blood pressure, reduces basically reduces your inflammation when you give an epinephrine, and then edema is gone. So that's basically what is treated for systemic hypersensitivity, immediate hypersensitivity. Subacute happens after an hour or two. Okay, so this is basically IgM and IgG, where you have your mismatched blood transfusion. So that's your cytotoxic type 2 reaction that you have, where it binds to all of your RBCs. Type 3 is where you have systemic lupus. The loop is where you have the antigen antibody complex, and that complex have no way to clear from your body, and that complex is what is causing cell damage in that particular part of the body. So that is your SLE, systemic lupus erythematosus. The T cells are your delayed hypersensitivity, and this happens after 1 or 3 days. So when you go to your TB testing in a hospital, they inject a little bit of toxins in your forearm, and then they send you home and tell you come back after 2 days, right? It's because it's a T cell reaction that happens over there. So TB testing is all delayed hypersensitivity, like poison ivy, like TB testing. These are all delayed hypersensitivity that we have. Developmental aspect of immune system. Now keep in mind when you are in the fetal stage, you do not have any exposure to any bacteria technically. Now they're contradicting that idea, but so far we think that it is in a sterile environment. But there are research that goes against it. It is again debatable. Um, so the first encounter of antigens that we come across is in the vaginal canal during birth, during labor. Okay, your entire immune system, even though it's developed in the liver and the spleen afterwards and in the bone marrow, even though you have all of those, they are never exposed to the antigen until after you're born. So the first year of your after you're born is very, very important for your immune system to develop into stronger immune system. The more you protect your newborn within that first year, neonatal stage, when more you protect that newborn, the more damage you're doing to the immune system, because the more antigen the immune system are exposed to, the more stronger your immune system, the less allergy that you're going to induce in your baby later on. I'm not telling you to take it out and then do what they usually do, like apparently they do like a lollipop thing for the chicken pox and all of those things. Don't do all those crazy stuff. But I'm saying don't be extreme and wiping down everything that the baby touches or in every surface. It has to be exposed to as much antigen. It is actually being recorded that if a baby is raised in an environment with animals, pet dogs or dogs better than cats actually, um, because they bring in all kind of antigen from outside, the baby born in that environment is actually healthier with less allergies than compared to kids born in a sterile environment. For your immune system to be strong, you need other things. You need vitamin D as well. So there is a new trend, in at least in COVID, that they're saying vitamin D is absolute important for COVID as well. So it's one of the preventative things that people are taking. They're taking zinc, vitamin D, and what else they're taking. So I think those things they're taking in order to prevent COVID and make your immune system stronger. Obviously, diet is everything. The rainbow color, you might have heard of that. Every color of the rainbow. So, as different colors of food that you eat, the better it is for your body because of all the supplement, the micronutrients that you're taking from different food products. So that is very important. Stress obviously is very, very important. The more stress you are, the brain basically is telling the body something is wrong. So the immune system starts to initiate a systemic inflammatory reaction. So you end up with a lot of cytokines in your body. When you have a lot of cytokines, you have a lot of inflammation going on. So inflammation can lead to GI tract issues like IBS, like Crohn's disease, like all of those inflammatory gastrointestinal diseases. It can cause diabetes, it can cause atherosclerotic plaque. So basically, stress is a leading cause for that, and also diet is also very, very important. The more processed food you put in, it's going to flag, because your 70% of your immune system is found in your GI tract, not in your lymph node, not in your blood circulating, but in your GI tract. So the more processed food you're putting in, those immune cells in your GI tracts are going to flag that these are unnatural products, and it's going to increase the inflammatory reaction in your body and cause a system-wide inflammation. So those are very important. As you get older, this is the problem. The thymus, where your T cells mature, they start to deteriorate as you get older. So because they're saying one theory is because the thymus is not there, your immune cells also are not exposed to different antigens, so the immune system dampens, which is what we're seeing in COVID as well, that because there's immune system is weaker in older people, they're tending to have more severe reaction in the COVID situation. It happens for every infection as well. As you get older, because the immune system starts to dampen up, because there's no thymus anymore educating the T cells, we're more susceptible to all kind of diseases. And you also have a susceptibility to cancer, to autoimmune disease, to immunodeficiency, all of those also seen as we get older because of that. It starts to fail as we get older. So this actually concludes your chapter 21 lecture, your immune system.